In this part of the spectroscopy experiment, you're going to be looking at something different. In other experiments, you've looked at what are known as emission spectra. These are the spectra that are produced when an atom is excited and produces light that we then look at with the spectroscope. In this experiment, though, we're going to look at something different. This is called absorption spectra. And absorption spectra are actually related to emission spectrum. Let's look at an example for lithium. From your lab handout, referring now to the spectrum that you see for the lithium emission spectrum, why, we can see that lithium, when it is excited, produces lines of light in these locations. These specific lines of light, and it appears as if we have a deep red, a green, a blue, and a couple of violets, consist of a fingerprint for lithium. If we see these lines in this particular pattern, we can be assured that we're looking at lithium's emission spectrum. It identifies lithium uniquely. A different type of spectrum, though, can be produced if we begin with a continuous source of light. In the lower spectrum, note that it appears to be almost continuous with some notable omissions. The color of light in the emissions in the continuous spectrum is often abbreviated by red, orange, yellow, green, blue, indigo, whatever that is, and violet. This is a nice way to remember the colors of light in the visible spectrum in order from low energy to high energy, or, if you prefer, in order from long wavelength to short wavelength. Roy G. Biv, R-O-Y-G-B-I-V, is the order of light colors in the visible spectrum. Now, if we take a source of white light, like a star, or our sun, or a white light bulb, and we pass it through a sample that contains lithium, the lithium very selectively removes from the continuous spectrum certain lines, as you can see here. Coinciding with the lines that lithium would be observed to produce in a different situation, we see that there is light that has been removed corresponding to deep red, green, blue, and the two violets. So we would say that the light that is missing from the continuous spectrum in that location right there has been absorbed. And we can see that there are other areas where light, I'm not going to label them all, where light has been absorbed, and they happen to coincide with where light would be produced if lithium had been excited by some external source of energy and asked to produce an emission spectrum. Now, the light that is not absorbed in the continuous spectrum, we would say is transmitted. And so we see in the absorption spectrum for lithium that yellow and green are for the most part transmitted, and other colors as well pass through the lithium sample with absolutely no ill effect whatsoever. So in our lithium absorption spectrum, what do we see? Well, we see a continuous spectrum missing specific lines that correspond to light that has been absorbed very specifically by our lithium. Now, this is the sort of effect that we often see with atomic spectra. Atoms in general are very specific about the color of light that they either produce or absorb, observed in either an emission or an absorption spectrum. Molecular species, on the other hand, are less discriminating, and rather than remove specific lines of light, they will often remove entire regions of a spectrum. Here from your lab handout is a very schematic view of the experiment that you're going to be doing today. And our white light source is simply a light bulb. And I'll show you that here in just a second. But what it produces is a continuous visible spectrum. And that light is shown through our sample which then has the opportunity to absorb certain regions of the visible spectrum, and then when we observe the transmitted light with our spectroscope, what we observe in that spectrum is a segment of the visible spectrum that has been removed. The missing or absorbed part of the spectrum, as you can see right here, is that center region that contained, for this specific example, whatever it happened to be, it looks like a fair bit of the green and yellow the green and yellow predominantly have been removed from the spectrum. These other colors have been transmitted here and on the left-hand side. So we see that light has been transmitted. When it's been transmitted, it has not been affected by the presence of the molecules, but a significant piece of the spectrum has actually been absorbed. Okay, so let's have a look at how this experiment might actually be done in the lab. Off here on the right-hand side of your screen, you'll see the light bulb that we'll use as a source of light. This light bulb has to be plugged into an outlet, as you can see here, and then to make it more convenient to do this experiment, we'll have you place the light bulb over on its side. It's actually sitting on the bench top right now. It will not damage the bench top. The bench top will survive this, so no worries there. But this is going to be our source of light in this experiment. Now, we'll also need a spectroscope in place, and so I have my spectroscope handy right here. Remember that the light source needs to illuminate, the light that we're going to study needs to go through the small rectangular opening here. Having the light bulb over on its side makes it possible for me to just place my spectroscope on the table. And now I can look into the spectroscope and adjust it back and forth until I see a very nice continuous visible spectrum with all of the colors of the rainbow. The alignment has now been performed. My next step will be to take one of these containers of food dye and place them in the path of the light. Now, just a quick review. This one, food coloring works in strange ways. This one is actually yellow, but it looks more orange. So that'll be the one that's marked yellow on your handout. And this one is red, and this one is blue, and the green one is most obvious of all. So what you'll do then is you will take and place one of them, or combinations, according to the lab handout, so that the white light from the light bulb passes through the green solution before it reaches the spectroscope. I do this without touching the bulb or the spectroscope to keep the alignment fixed. One way that I find works quite well when you're doing this experiment is to look into the spectroscope and to remove the dye and then place the dye back in position again and remove it back and forth, and you get a little light show as you'll see the continuous spectrum and then the absorption spectrum. It helps make things more clear what segments of the visible spectrum are being removed and what segments are being transmitted. Okay? Then you'd move on to another container and combinations as per the laboratory instructions. Let's take a look, though, at what this might actually look like. Down here, I have taken some pictures using that very spectroscope to show you what one experiment might actually look like. Now, it's pretty obvious that the upper spectrum is the continuous spectrum that we observe with our spectroscope with no sample interfering. And we have all of those colors that we expect to see. Let's see here, we've got red, orange, yellow, green, blue, indigo, and violet out here. And then as we position a sample between the light bulb and the spectroscope, we see something similar but quite different. You'll notice that we still get a very weak amount of red. This would be light that is transmitted. And then there's a whole region of the spectrum that is gone because it has been absorbed. So the molecules are very busy absorbing these wavelengths of light and probably converting them to heat energy. And then we get out off to the right where we see, although it's dimmed a wee bit, we see the remaining colors, the green, the blue, the indigo, and violet, which are also transmitted. Okay. In your lab notebook, what we would like you to do is to record those parts of the visible spectrum that you actually see. You're going to be asked to report the colors that have been transmitted in your lab notebook. And so in this case, I would say that red, green, blue, indigo, and violet have all been transmitted, and anything that I left out, namely the orange and the yellow, have been absorbed. And that's it. You'll experiment with different combinations of food coloring and water, recording only those colors that you observe.